Hello, everyone. It's Patrick Morrison from the Wolfpack Astrobiology Podcast, and in this short video, I'm going to give a little rant about de-extinction. For those of you who don't know, the creature depicted here is the Tasmanian wolf, also known as the thylacine or Tasmanian tiger. Despite the name, it wasn't a wolf or even a member of the dog family. It was a marsupial, and the similarities are a textbook example of convergent evolution. When European settlers arrived in Tasmania, they thought it was killing their sheep, so they relentlessly hunted it, which led to its demise. On the 7th of September 1936, the last one died in Hobart Zoo. Its extinction was so recent that you can see some old black and white footage of living animals. Today, many people deeply wish it hadn't been driven to extinction, and in 1999, a project was launched by the Australian National Museum to resurrect it. How did they attempt this? Well, one of the joeys was preserved in alcohol, so they sequenced bits of DNA from its tissues and attempted to assemble a complete genome. This would then be inserted into the egg of a Tasmanian devil, which would then carry the thylacine. Regrettably, the attempt failed, but in my opinion, the research team jumped the gun trying to resurrect a species whose closest relative branched off from it over 30 million years ago. In my opinion, if we truly want to resurrect extinct species, we should hone our craft with a series of incremental steps. First, I think we should attempt genetic rescue using museum specimens. To understand what genetic rescue is, you first have to understand what the extinction vortex is. This is where, as the result of a drop in the overall population size, the genetic diversity decreases, resulting in inbreeding depression within a population or species. This leads to fewer individuals surviving to reproduce, so the next generation has less genetic diversity, inbreeding depression gets worse, and the cycle repeats. When a population is in the extinction vortex, oftentimes individuals from another population are introduced to increase the genetic diversity. However, there are some limitations to this approach. First is that since you're introducing individuals from another genetically distinct population, you aren't exactly preserving the original population. Second, oftentimes there is only one population left in an entire species. One way to circumvent these problems would be to take preserved samples from museum specimens, sequence the DNA, and identify alleles no longer present in the population. These can then be inserted into fertilized egg cells or zygotes, which would then be implanted into a female. This way, we can not only improve genetic diversity, but retain the original genetic composition of the population. One animal this could help is the black-footed ferret. At the turn of the 20th century, it nearly died out due to the disappearance of its food source. It is recovering, but problems relating to inbreeding, due to having such a low surviving population, have hampered recovery efforts. Reintroducing genetic material from museum specimens can help mitigate these problems. So, the first step to de-extinction would be to resurrect extinct alleles, but from there, we can move on to the next step, resurrecting extinct populations. This is where a genetically distinct population inhabiting a given area has been driven to extinction, while other members of the species survive elsewhere. Whenever you obtain DNA from museum specimens, you'll get a jumbled bunch of fragments, which you then have to assemble into a complete genome. This is much easier to accomplish when you have a reference genome with the same genetic structure, i.e. the same chromosomes, which have the same order of genes on each chromosome, and other living members of the species can provide this reference genome, allowing you to assemble the genome by aligning sequences with known positions in the reference genome. Some populations that have recently been driven to extinction that we could use this approach with include the Japanese wolf, which, unlike the Tasmanian wolf, was actually a wolf, and the quagga, a subspecies of zebra. The third step would then be to resurrect a species closely related enough to an extant species that the two could reproduce, but the resulting offspring would be sterile. This would be more complicated than the previous task, since the genome you're trying to resurrect and the reference genome can have some subtle differences. But at least you know that the surrogate mother, who is of the related species, could carry the offspring. One of the problems with this step is that it would involve a lot of guesswork, since we most often won't know who can and can't produce offspring with whom, and if a species is extinct, we can't exactly try to breed it with a related species to find out. There may be archived documents from zoos indicating that a now extinct species can breed and produce sterile offspring with a living one, but one indicator we can use is phylogenetic bracketing. Basically, if you have an extinct species and two living ones, where the living ones can produce sterile offspring, and one of the living species is more closely related to the extinct species than the other extant one, it's a safe bet that it can act as a surrogate and provide a reference genome for the species you intend to resurrect. One species that could possibly be resurrected by this approach is the Caribbean monk seal, using the Hawaiian monk seal. From there, we can home our craft and resurrect more and more distantly related species from living ones from more and more fragmentary material. And then, who knows, maybe one day we'll have Tasmanian wolves and even mammoths roaming around once more. So, that's what I have to say about the topic, and if you have another view on the matter, I look forward to reading your comments. Thanks.